Welcome to the BIRCH Bioinformatics System. My name is Brian Frostensky, and today we'll take a look at how BIRCH applies the principles of data science to get more out of your BLAST searches using databases installed on your own computer or remotely at NCBI. Most of you watching this video have done BLAST searches through the NCBI website, and although NCBI has a powerful cluster of servers, this is offset by the fact that NCBI processes literally hundreds of thousands of searches every day from users worldwide. It's often the case that you'll get a faster turnaround on BLAST searches done on your local computer. More importantly, I'll demonstrate the advantages of running BLAST through BioLegato. BioLegato organizes data and tasks according to object-oriented principles, putting hundreds of bioinformatics tools at your fingertips and empowering the process of discovery. Think of BioLegato as a family of applications that run other programs. There are many BioLegato applications, each specialized for running tasks on specific types of data. Every time we open a BioLegato application in a new window, we refer to that BioLegato window as an object. For example, here we see BioLegato objects for handling BLAST hits, multiple sequence alignments, doing keyword queries at NCBI, or handling sequencing reads for genome or transcriptome analysis. When we run a program from one BioLegato object, the results are sent to a new BioLegato object, appropriate for working with the output of the previous step. This in turn lets you discover new things from the results and go on to the next step. We call this process ad hoc pipelining. Let me show you how object-oriented BioLegato objects fit into the larger context of data science. The field of data science exists in part because data is messy. The hard part of working with data is often the laborious pre-processing that has to be done before you can begin the analytical steps. This slide summarizes much of what data scientists do, which is best thought of as a pipeline. You can see we're starting at the top with disorganized data, doing some steps on it, maybe sorting or culling, arranging the data to make sense out of it in a unified structure, and then finally presenting it in some way that makes sense out of it. So all of these steps occur in what we might refer to as a data pipeline. The early steps are data cleaning, doing things to get rid of what data we don't want, maybe false positive BLAST hits, for example, and then the analytics later on to learn things and inform our next steps. One of the unifying threads in the BioLegato paradigm is that the user should always be able to use the output from one step as input for the next step. BioLegato automates the flow of data from one step to the next and makes it as painless as possible to inspect and clean the data and make decisions at each step. We call this ad hoc pipelining. For example, BL Protein is a BioLegato application that runs tasks on protein sequences. Today we'll see how BL Protein makes it easy to compare protein queries against protein or DNA databases. In a larger context, BLAST searches are one step in a more complex data pipeline. So aside from the mechanics of running BLAST, I want to show you how BioLegato orchestrates all the steps in that pipeline. Here's a typical BLAST report, which is human readable but not really machine readable. Usually you want to do something with your output and the sequences that you find. We refer to that process of discovery as downstream analysis. Remember that we want to identify the hits that we do want and get rid of those we don't want. So to keep the results machine readable, a second copy of the results pops up in a BioLegato spreadsheet called BLP Fetch. Spreadsheets are powerful because the human eye is very good at scanning through maybe hundreds or thousands of results. BLP Fetch makes that easier by letting you sort results based on things like the sequence title, the e-value, the length of the alignment, and delete, and finally create smaller data sets that you actually want to retrieve. So once you've selected the hit lines that you want, you can run seek fetch, which will then go to NCBI and retrieve the GenBank entries for the sequences that you specified. The output appears in a new BioLegato object, in this case a BL protein object that lets you do further steps on the proteins. So all of these downstream analysis steps are at your disposal once you've retrieved the sequences. So let's begin our demo now and see ad hoc pipelining at work. As an example, Let's start with the amino acid sequence for the SARS-CoV-2 spike protein, which is the best potential vaccine target. Yuan and colleagues have used X-ray crystallography to determine the 3D structure of the spike protein when it's complexed with a neutralizing antibody from a patient infected with the SARS virus from 2007. In panel B, we see the antibody heavy chain in gold. The 2007 protein has an alanine at position 372, which creates an N-glycosylation site at position 370. Mutation to a threonine in the 372 eliminates the N-glycosylation site. We can see in panel C where the N-glycan would fit into a groove in this protein. In this light, it would be instructive to compare spike proteins from a number of SARS strains for a more thorough understanding of amino acid conservation at these positions. In 2D, we see that the antibody binding may also be affected by substitution of the alanine at position 384 in the 2007 protein with a proline, which is likely to result in conformational changes as well. From an experimental perspective, it will be useful to know whether these mutations are very recent or whether they have been present in distinct strains of SARS for a long time. For that, we need to compare our two spike proteins with spike proteins from other SARS strains in a multiple sequence alignment. The best way to find those sequences is to use these spike proteins as queries in BLAST searches. We can retrieve the two sequences from this paper using the accession numbers found in the manuscript. So to get started, let's launch BIRCH. And this is the BIRCH launcher, which in turn lets us launch other BioLegato applications. So one of those is NCBI. So I'm going to launch NCBI, which is a program for retrieving sequences from NCBI using keywords. In this case, the keywords that we want to use are going to be the two accession numbers. So the accession numbers are going to be inserted into the protein query tool that allows us to create search terms. So you can set search terms like the author or the gene name or the protein name or a text word. In this case, we'll set the accession field and then copy accession numbers from the paper. So I'm going to copy the first one and then copy the second one. And of course I've got to set this other field to be accession. And since I want this to be an either or, I want queries that match either of the two accession numbers, I have to put an or term in here. So now we're ready to run the retrieval. I just click on the output button and output comes up in another NCBI window. So we can open up the title column here and we can see that these are the ones we wanted. So the next step is to just select the accession numbers and use protfetch to retrieve the protein sequences. So that comes up in a new BioLegato window, which is exactly what we want to do in ad hoc pipelining. One of the fundamental ideas in data science is to learn about your data as you go. So at this point, I think it's a good idea before we do our BLAST searches to learn a little bit more about these proteins. So the first thing we can do is select both names and then run the protein statistics program, which just gives us a little bit of information. So we can see here that they're both about a little over 1,200 amino acids and about 140 kilodaltons each. These are big proteins, and they're also somewhat acidic, and so we have an idea about some of the characteristics of these proteins. So it's also probably a good idea to see how similar they are, so I'm going to run S-search, which is a rigorous Smith-Waterman alignment that will tell us something about the similarity of these two sequences. So we can see here that they are about 76% identical and 91.5% similar. Now the difference is that similarity is taking into account conservative amino acid replacements. So the conservative replacements contribute more to the score than more extreme replacements. And so we have an alignment here which shows us conservative replacements and somewhat less conservative ones, and then finally gaps which are inserted by the program to make similar amino acids line up. So because there are some differences, we stand a good chance of retrieving different hits with one sequence than with the other. So we'll probably do both sequences as BLAST queries one at a time and then combine the hits to create our data set. So we'll do BLAST searches for each of these sequences, and that's done from the database menu. So you'll see here, of course, we've got the ability to send BLAST searches to NCBI, and we also have the FASTA programs that are from Bill Pearson, an alternative to BLAST that can be used to search local databases. But today we'll use BLAST P against the local UniProt database. And so there's the menu for BLAST P, and it's worth just going through some of the options. Of course, first of all, you've got the choice of databases, and so this lists only the databases that are installed on your system. So the list might be a lot bigger depending on which databases you choose to install. The default here is UniProt, so we'll stick with that. You have a choice of several different BLAST programs, a number of scoring matrices, and then the E-value. So just to make sure everyone knows what the E-value is, it's the number of matches that you would expect to see by random chance when you look at a given hit. In other words, if a hit has an E-value of 5, then it's probably not significant. That would mean that you would expect to see as many as 5 hits that good just by random chance. But if the E-value is an extremely small number, like 10 to the minus 11th that we see here, that means that the chance of seeing a match that good is only going to be possible 1 out of every 10 to the 11th times, and so you can say that's probably a significant match. Now, we have the ability to use multiple cores, and so a typical PC from the store will have anywhere from 2 to 4 CPUs or cores. This machine has 8 of them. The default is to use 1/4 of the cores, but since I'm the only person using this machine, we'll use 6 cores. And then we have several other tabs here. You can see we can restrict the search based on taxonomy or keywords. We have a number of different scoring schemes that can be used. Queries can be filtered, so the default is to not filter out low complexity regions, but you can do that if you want. And then finally the output. You get to choose how many alignments you want to see and where the output goes. By default it goes to a web browser, and also the table goes to BLP fetch. You could send those, for example, to files if you wanted to start the search and then come back later. But we'll go with the defaults on that. And so now we should be ready to run our BLAST search. So I've zoomed out now to do that, and I click on Run. We're doing this in real time because UniProt is a small database, so we should get our results almost right away. And so we'll just wait for the web browser to come up with the output, and then we'll see what that looks like. So there is the output in HTML form that we've talked about before, and in another moment we should get the summary of the hits in a BLP fetch window. So just taking a quick look at the output in the report, of course for every hit you've got a link to the sequence and then a link to the alignment that's shown here. So we get all of that information, and then we get much of the same information in the NCBI report. So we can see here the subject title. Each hit is the subject sequence. There are a total of 70 hits that were found, and maybe I should just zoom in on this to make it a little easier to see what we're doing. Um, the scientific names give us a little more information. You can see that we've got a collection of sequences from many different strains of COVID that attack different organisms. Interestingly enough, I didn't know that this virus would cause hepatitis in mice, for example, and so that's an interesting observation. Of course, we have the taxonomy ID, which gives you much the same information. This is the ID of the virus, not of the host. And then if we move things around a little bit, we can also see the alignment length. And so that might be an important part of our search, because you can see here some of them are the full length of the alignment, and then others are quite small. 195, for example, there means that probably the subject sequence was not that similar. If you have a look over here, you'll see that the subject length is full length, but only 195 bases of that hit are in the alignment with our query sequence. So we're probably not interested in things right now that only match part of the query. And so that will be one of our criteria for deciding on which sequences to look at. So if I know I only want the long ones, then I could sort on column F, and so that's done by selecting all and then using BL sort to sort on the sixth column, and we'll choose descending as the order, so that when the output comes up in a new window, we will see that in the order that we want. And so I'm going to spread this out a little bit, and then subject length again is column F. Now you can see that we've got all of the ones that are the length that we're interested in, and so I'm just going to select those that give us a full length alignment. So for the first query, which is our 2020 spike protein, that will be the set of sequences that we choose to retrieve. And in fact, what I'm going to do is I'm going to now extract that into a separate PLP fetch window, so that I can have a little better handle on these things. You'll note here, by the way, that we've gotten an interesting zoo of sequences. We've got a bat sequence, and I guess a human one, maybe 4 bat sequences, and then the murine hepatitis sequence. So we've got an interesting mix of sequences, and we'll use that in our next steps. So I've closed some of the windows, and I've put our first list of hits from the first query over to the side, and now we'll do the second search and get the results from that. So use all the same parameters, and once again we'll get the output both in an HTML window as well as in the other windows. So now I'm just going to minimize this report to get it out of the way, and now have a look at the same window from our second search. You can see there are 68 hits found, and if we take a look once again at the alignment length, you can see here there are a fair number of long alignments, and just taking a look at what we've got, again, it's from a variety of different species, and so I want to make sure I get all of the long alignments. So again, I'll select and sort in descending order on column F. Get our next window, and there are all of the alignments that we wanted. So now I want to get both sets of these sequences into the same window for retrieval. So what I'm going to do is select the ones that give us the long alignments, let's say just up to anything larger than 1200, and we can use the copy out command to copy these hits out to a clipboard, and then now we go to our first set and we paste those in. So if we open up this window, you can see we've got both sets of hits, and there are some duplicates like this one and this one are duplicate. That's not a problem. Those will be eliminated before the hits get sent to NCBI for retrieval. So now we're ready to retrieve our sequences. We select all, we open seqfetch and retrieve those sequences from NCBI, and now with ad hoc pipelining, of course, they go to a new BLP or BL protein window. So again, what I want to do is I'm going to I want to have all of these query proteins together with the hits, and so I can copy these out of the first BL protein window and paste them in to the second window, and now we have all of our hits and the original queries in the same BL protein window. So at this point, it's a good idea to compare the way we would do the same blast search on the NCBI website with what we've already done. And so if we go to the output, the first page of output that we get is a table of the hits, and this looks superficially like the BLP fetch output, but this is a static table. It's not a spreadsheet. You really can't do much in the way of operations with it. You can do things like selecting with checkboxes individual sequences or deselecting them. You really can't do any kind of sort or anything like that, and then you can only look at a maximum of 100 hits at a time. And so it's a little bit limited compared to what you can do with BLP fetch. The graphic summary is actually quite a good form of output. We see here what we pretty much learned from looking at the tabular output, and that is that the top hits with the highest E values have alignments that span the entire length of the query sequence, and then the rest of the hits only span maybe the C terminal half of the alignment or some blocks in the N terminal half. And so the ones we were most interested in were these longest ones. The alignments page gives you the alignments much the way we would see it in the output that we looked at from the BioLegato search. And then finally, the taxonomy tab gives you the results organized by the different bacteria, or I should say viral strains. And so once again, it's a static page. We don't have the same capabilities for working with the data as we do with the BioLegato output. So for example, if I wanted to group by the viral strain, I work with the subject taxonomy ID column, so that's the fourth column or column D, and sort based on that column, and then get the results back in sorted form. So we might want to just zoom in on that and have a look at how the output is grouped now. So for any given strain, all of the hits for that strain are grouped together, and so that lets us have a little more fine control over what exactly it is we want to retrieve. So at this point, I think it's best if we go back now to the hits we retrieved and take a look at some of the downstream analysis that we can do. So to do our multiple alignment, we just have to select all of the proteins in BL protein, and then in the alignment menu we run MAFT, which is going to be using the fast multiple alignment that's the default, and output goes to a BLP align object, which is the BioLegato for multiple protein alignments. Yuman and co-workers demonstrated that the antibody binds to the cyan-colored regions in this protein, and you'll see that there are 4 asterisks which indicate the sites that are polymorphic between the SARS-CoV-2007 protein and the 2020 protein for SARS-CoV-2. Let's go to the first of those sites. So here once again we have the 2020 protein with an alanine, and all the rest of them except for a very distant bat sequence have a threonine at this position. So these 2 are human proteins, and these 4 over here are bat proteins. So this alanine in the 2020 protein appears to be a fairly recent evolutionary event. It's interesting that at this position the proline in the 2020 protein is more bat-like, because all the other bat proteins have the proline, whereas the other 2 human proteins have an alanine at that position. So there seems to be a sort of different evolutionary pattern at that position. We might be able to learn a little bit more about the conservation of amino acids at these sites by looking at a sequence logo of the protein. So to create a sequence logo, again, we select all of the proteins and then run web logo to create our sequence logo, and then that will give us a better view of the amino acid substitutions at each position as measured in information content at each site. So now we have popping up the sequence logo for the entire set of proteins in the alignment. So the important thing to know about sequence logos is that the y-axis gives you information content at each position measured in bits of information. So one way of thinking of bit scores is as a measure of the deviation from randomness. So high bit scores are associated with a strong conservation of amino acids, and conservative replacements result in a moderate decrease in the bit score at a given position. More drastic replacements, of course, will give you a much lower bit score at a position. So I think it'll be easier to talk about this output if we have a slide where the critical amino acid substitutions are highlighted. So here's the same position we were looking at just a moment ago, where we have most of the bat sequences with threonine and the more recent amino acid replacement of an alanine in the human sequence. And then here's the proline alanine substitution that we saw. One thing we're starting to see is that surrounding these substitutions are regions of higher information content and therefore higher conservation, and somewhat lower conservation at these polymorphic sites. Here's another important site in where we had a threonine to methionine mutation. Threonine is the bat-like amino acid. Methionine is the one in the 2007 human protein. And then at 559 in the alignment, we have asparagine in most of them, which is a bat-like amino acid, whereas you have a histidine, the very small letter H there, in the 2020 protein. Yet another way of examining a multiple alignment is to export the entire alignment out to JAL view, which is a very comprehensive multiple alignment viewer and editor. So we have a lot of different ways of viewing the alignment in JAL view, and just to keep it brief, I'll mention the conservation graph at the bottom. So the conservation graph indicates the degree of conservation at a given position. So as in keeping with what we were looking at before, position 412 is somewhat less conserved than the surrounding amino acids. So we see here again the AT polymorphism in this protein. As well, the PA polymorphism at position 424 in the alignment once again is at a local low point, shall we say, in terms of conservation. So I want to emphasize that really what we've been working with so far is a very small data set, and in a research situation you probably would want to work with a bigger data set and probably make some subsets of that data set to really dissect what's going on. So here is a search of a larger database, the non-redundant database, in which 527 hits were found using the 2020 protein as an example. So we can see a number of things. First of all, there's actually quite a large number of hits, and a very large number coming from the Wuhan seafood market, so that means the data set is going to be a little bit skewed. Also note, we can see here by examination a synthetic construct, so we'd like to get rid of things like that from our data set. Maybe a good thing to do would be to select all and then to sort the data set based on the scientific names of the subjects, and then we will do that. I suppose it doesn't matter, we can do it in descending order, alphabetical order, and then we get a new one popping up, and right away that helps us because we can see that there are several other synthetic sequences or recombinant sequences that we'd like to get rid of. So I'm just going to get rid of those right now and not have to worry about them. So from our original data set of 527 hits, we can create some subsets that will let us examine in more detail the animal transmission of this virus. So here's the complete data set, and then of course this is a bat virus initially, and then in the 2007 SARS was transmitted through civets. So here's our civet data set, and it is thought that the 2020 virus was transmitted through pangolins. So we have a pangolin data set, and then finally presumably from pangolins the virus got to humans, and then here would be our human data set. So the ability to create these database subsets very easily lets us test any number of hypotheses about the origins of these viruses. We've shown that desktop tools let you work more directly with your data and let you get more out of your data than web applications. These examples have been done using locally installed copies of BLAST databases. If you'd like to install BLAST databases on your computer, see our video on installing BLAST databases found on the BIRCH YouTube channel. The video will take you through how BioLegato can help you make decisions on which databases to install. From there, installing the databases can be done with a few mouse clicks. We've also shown how BIRCH BioLegato applications implement the principles of data science, providing an organized framework for managing your data and giving you the tools to learn from it. Ad hoc pipelining gives you a framework for experimenting with your data and asking the what-if questions. Finally, I want to emphasize that BLAST is only one part of the larger BIRCH system. BIRCH seamlessly integrates bioinformatics tools through BioLegato. The object-oriented applications of BioLegato include basic sequence tasks, sequence alignment and phylogeny, genome and transcriptome assembly and visualization, and comparative genomics. That concludes our BLAST tutorial. I'd like to thank, first of all, the authors of BioLegato, Graham Alver and ABL Roche. I'd also like to thank the agencies that made this work possible: Genome Prairie, Manitoba Innovation Energy and Mines, and the University of Manitoba. If you'd like to install BIRCH on your own system, you can download it at the website shown. BIRCH is free open source software, but remember, you pay for open source software by citing it in your publications. Without open source software, our research would be impossible to do. Thank you for watching, and visit our BIRCH YouTube site for other videos on bioinformatics using BIRCH.